Hello, my name is Glenn Pierce. I am the Vice President for Medical for the World Federation of Hemophilia and have had a long-standing interest and involvement in gene therapy for hemophilia. Today we'll be talking about other strategies and targets for gene therapy in the treatment of hemophilia. Our objectives really will be to look at other approaches besides adeno-associated virus and to look at some of the pros and cons of adeno-associated virus throughout this talk. Gene therapy got its conceptual start nearly 50 years ago now with an article that Ted Friedman wrote in Science wondering about gene therapy for human disease. This wasn't that long after the identification of DNA and RNA and their functions, and he raised the question of difficult scientific and ethical problems, much as we're raising some of those same questions today about gene editing. If we fast forward now nearly 50 years and ask the question, why AAV vectors for hemophilia gene therapy? Well, the bottom line is because they work. But that has evolved over a period of 25 to 30 years with many failures along the way. By the time we've reached this point, they are the most viable approach toward giving therapeutic benefit to individuals with hemophilia, both factor VIII and factor IX deficiencies. Overall, they've got a good safety record and have been used in a number of different disease states in clinical trials. There are many issues that we'll be discussing, including the variability and durability of responses, the fact that perhaps up to half the people have pre-existing immunity to different AAV serotypes. Serotypes are AAV particles that are closely related to one another but have evolved enough away from the primary AAV that they can infect different tissues in the body. So we have used a number of those AAV serotypes for gene therapy, and they all have some evidence of prior exposure in a number of people, which has induced antibody responses that prevent their use in those who are positive. This immune response that we see before administering the AAV is amplified considerably after giving the AAV, which precludes redosing. Very high antibody titers result from administering the AAV gene therapy. AAV is one of the smallest viruses that can infect humans, and as such, even after removing the AAV genes, there's not much room. The B domain deleted factor VIII gene, for instance, barely fits into the AAV, so there are size limitations as well. AAV can produce a little bit of random integration that's not well defined, not well studied, and raises a question about long-term safety. And then the final concern about AAV is that we are administering trillions of viral genomes to patients, but a very small fraction of that winds up producing proteins that get secreted and are useful for therapy. All of that said, the AAV is a working virus vector that can give us a therapeutic effect in hemophilia. What about solutions for improving AAV vectors? As we have looked at all of these issues that surround the use of AAV, many solutions have been suggested. Most have not really been tested, though. This is an important component, not only as clinical trials progress toward phase 3, but also as new trials are established that may show improvements of AAV vectors, transduction efficiencies, and the like. So all of these potential solutions are being studied, but not quite at the pace that they will make their way into this first generation of AAV gene therapy that may be approved as early as 2020, 2021. If we look at AAV vector transduction of hepatocytes, looking at this cartoon, what we can see in number 1 is that if you have antibodies to AAV from a pre-existing natural infection, that will prevent it from even getting to the target cell. And so no gene therapy efficacy will be achieved. In the absence of antibodies, number 2, the AAV can bind to specific cell surface receptors and get endocytosed through the usual mechanisms of receptor transcytosis. Once it's in the cell, it may encounter some innate immunity because, after all, our cells are designed to defend us against viral infection. And that's what this can appear to be like when it is being used as a therapy. But then enough AAV may escape such that it gets uncoated in the cytoplasm. The DNA goes into the nucleus, where eventually it forms episomes, which are stable, closed loops that can use the host machinery to transcribe and translate protein, which can then be secreted. The capsid itself goes into the endoplasmic reticulum, where it encounters major histocompatibility class 1 antigens. Some of those may be able to bind specifically to capsid peptides, in which case they are distributed on the surface of the cell where they can stimulate specific anti-capsid cytotoxic CD8 T cells. That then may result in the destruction of the cell containing the transgene. So we're dealing with issues in efficiency, variability, durability as a function of all of these complex steps that occur within the cell. What can we do about pre-existing immunity? Well, some groups are investigating in animal models immunoadsorption. That involves using a column, such as a protein G or a protein A column, that can adsorb out immunoglobulin, IgG, from a patient who may have high titers of anti-AAV. That's a transient effect. It doesn't last very long, but it may last long enough and drop the titers sufficiently such that one can administer the AAV and deliver the transgene to the target genes. The work is ongoing here to look at the efficiencies of removal of the IgG, since in a number of patients, significant amounts, logs worth of IgG will need to be removed in order to allow for targeting to the hepatocyte. Then there's the question of intracellular toxicity after AAV delivery. What we recognize is that in a number of patients, elevations in liver transaminases are seen. They're transient, they're not very high in most cases, maybe 1.5 to 2x above normal in most of the cases. However, they are of some concern because liver transaminase elevation does indicate hepatocyte death. What are the possible causes of liver enzyme elevation? Well, there are 3 hypotheses, and it may be one or more of these that has an effect on the hepatocytes in individual patients. First off, it's the degradation of a very high load of AAV capsid. A lot of capsid goes into the cells because a lot is administered intravenously, and the cell has to digest that capsid. It has to degrade it back into its constituent amino acids, and that takes a significant amount of energy for the cell to do that. Then there's the cytotoxic T cell problem that I've just described. And the third possibility is what's known as an unfolded protein response, and that's more specific to factor VIII. What's been established for a number of years now is that it's very difficult for cells to make the factor VIII protein. It is a complex protein, one of the largest in the body, and even if we're using the B domain deleted factor VIII, it's been shown in cell cultures that it's very difficult for cells to make, and it can cause toxicity within those cells. That toxicity can result in cell death as well, and of course, loss of factor VIII protein expression. So these are all the areas that need to be investigated, but yet are not being sufficiently studied to help us get to some of the answers to these questions and then develop potential solutions to these potential problems. So this shows an example of the elevation of transaminases within the liver on the upper left graph, and it shows the coincident development of CD8 cytotoxic T cells that are specific for AAV capsid, which induces death and destruction of the cells. What we see on the bottom are the various immune responses that can occur in response to transduction and transgene expression. What we see are CD8 T cells that can attack the hepatocyte containing the transgene. We also see that CD4 T cells, helper cells, can be activated, and those are important in generating a significant antibody response to the AAV capsid, precluding subsequent readministration. So there's almost no effort to investigate these three critical questions involving the capsid, cytotoxic T cells, and the unfolded protein response. Much more work is needed in these areas to better define that in individual patients and groups of patients. What are some alternatives to AAV? Well, a lot of work has been done with both viral and non-viral alternatives. The most advanced virus that is being used in hemophilia is lentiviruses. Lentiviruses are derived from HIV. They are missing a number of HIV genes, and the factor VIII or factor IX gene can be inserted, just as it is for AAV. Work is being done both ex vivo with hematopoietic stem cells and in vivo, specifically targeting hepatocytes, utilizing lentiviral delivery of both factor IX and factor VIII in a number of preclinical models, including large animals. A different approach involves not using viruses at all, which would be an ideal situation. Lipid nanoparticles have been in research now for 30+ years to deliver nucleic acids, such as DNA. I talked about how inefficient AAV is. These are even less efficient at putting genes into cells. Nonetheless, immune responses are not generated to these lipid nanoparticles. Therefore, repeat injections may be possible. If a patient doesn't have a high enough level of factor VIII or factor IX, one may be able to give this repeatedly in order to generate that kind of a level that would be therapeutic. Another approach that really is the future of gene therapy involves gene editing, and it involves adding the gene directly into chromosomes. It could replace an existing gene. In the case of hemophilia, it doesn't necessarily need to do that. It can just be added into the chromosome. In some cases with hemophilia, if there's a specific mutation, it could even repair the endogenous gene. Gene editing represents the future of gene therapy, as I said, because it allows for permanent correction of a phenotype. Once it's in the chromosomes, it will continue to express protein for the life of the cell. If the cell divides, it will be found in both daughter cells as well. So it's an attractive approach, not quite ready for human clinical trials, but under study in preclinical models for hemophilia. The final approach is cell therapy. With cell therapy, a number of different cell types have been used, both stem cells as well as hepatocytes, to deliver the factor VIII or factor IX genes directly to individuals with hemophilia. This slide shows a diagram of lipid nanoparticles delivering nucleic acids. Most of the work done here is with small RNA molecules, such as inhibitory RNAs, but potentially genes can be incorporated into these lipid nanoparticles as well. A lot of work has been done to make them non-toxic to cells and to allow them to target, for instance, to hepatocytes. And so we look forward to further work in this area. It's not quite ready for clinical trials yet, but it's moving in that direction. We need to identify ways of increasing the efficiency of gene expression utilizing these. They offer a number of advantages over viral vector delivery. What is a key difference between lentiviral and adeno-associated virus, AAV, vectors? Key differences between lentiviral and AAV vectors are shown on this chart. They come from different families. In one case, they're enveloped with a lipid envelope, such as lentiviruses, and in another case, they're non-enveloped, just showing protein that can interface with receptors on the cell surfaces. Packaging capacity for lentivirus is twice that for adeno-associated viruses, and as I mentioned, the lentivirus integrates into the host DNA, while the AAV mostly does not. It remains extra chromosomal for the most part. They both can transduce both dividing and non-dividing cells, And as I mentioned, the lentivirus gives a permanent correction, whereas the AAV in the form of episomes may be lost with cell division. If we look at the comparison of gene transfer with lentiviral and AAV vectors, it's really very similar. It's a virus that can get into the cells through specific receptors in both cases. In both cases, the DNA or the RNA in the case of the lentiviruses needs to get into the nucleus, where both of these utilize host machinery. The lentivirus utilizes the host machinery to make DNA from the RNA and then integrate that DNA into the chromosome. The AAV utilizes host machinery to take the DNA, which is single-stranded, turn it into double-stranded DNA, have it form an episome or a circular piece of DNA where RNA can then be transcribed to make protein. So both approaches can work and provide therapeutic levels of factor VIII, at least in animals, and in the case of AAV, in humans. Genome editing for hemophilia includes approaches that are in vivo or ex vivo. Let's look more closely at gene editing. With gene editing, which is a relatively new technique and technology that's been established over the past 5 to 8 years, we are able to insert a new gene directly into the chromosomes using specific bacterial enzymes that can open up our DNA and allow the new gene to insert itself. This provides for a permanent correction within the chromosome. If we look at one specific method of gene editing, such as CRISPR Cas9, what we can see in this case, which is an abstract from a presentation given at the ISTH meeting in July of 2019 by Alan Brooks, it shows that he's able to cut the DNA of the host, in this case hepatocytes, and insert the B-domain deleted factor VIII gene within the chromosomes of the hepatocytes and produce mRNA, messenger RNA, and secreted factor VIII. This was done in animal models and has shown levels of factor VIII well within the therapeutic range. If we turn now to cell therapy approaches, a number of groups have used both stem cells as well as mature hepatocytes to deliver the factor VIII or factor IX genes. In this case, shown in the cartoon, the factor VIII gene is put into a lentiviral vector. That lentivirus vector is able to transduce mesenchymal stem cells that can be taken from the host. And those mesenchymal stem cells can be induced to differentiate into a number of different cell types, including cells of the bone marrow as well as hepatocytes, and thus can potentially make the factor VIII within those differentiated cell types. This can be done in an autologous way, meaning that the stem cells can be taken from the patient and manipulated ex vivo and then returned with the factor VIII gene back to the patient where they can secrete factor VIII protein. This has been done in animals and actually has been done in humans, not fully successfully, but a paper came out nearly 20 years ago from Roth and colleagues that showed that they could remove fibroblasts from a skin biopsy, grow those in vitro, add the factor VIII gene, and then reimplant them into the peritoneal cavity. They think they saw a little bit of factor VIII activity for a short period of time in the circulation, but never pursued this to the point of achieving therapeutic benefit for patients. In this next approach, endothelial cells can express factor VIII. This is the natural cell type that does express factor VIII, the sinusoidal endothelial cells of the liver. And what can be done is to pull autologous stem cells, induced pluripotential stem cells from a patient's circulation, transduce the factor VIII gene using a lentiviral vector. These cells can then be differentiated into endothelial cells, can be delivered back to the patient, and rescue the hemophilic phenotype. The conceptual benefits of this are very clear. It's very simple. It's more challenging when we reduce it to practice or attempt to reduce it to practice. But nonetheless, this has been done in animals and has provided for therapeutic levels of factor VIII when animals receive these new cells. And then in a final set of experiments, hepatocyte-like cells can be generated from induced pluripotential stem cells. And the approach there would be to take peripheral blood mononuclear cells from patients, reprogram them using transcription factors, potentially small molecules, into induced pluripotential stem cells, do gene editing to put in the factor IX gene, direct the differentiation toward hepatocyte-like cells, and then reimplant those into the patient where they home to the liver or could reside in the peritoneal cavity and produce substantial amounts of therapeutic factor IX. This has been accomplished in mice. And so with all of these approaches in cell therapy, we've got some reasonable results that have occurred in animal models, and it's a question of whether this transition can be made to human clinical studies. No human clinical studies are currently planned in the short term at this point. More work needs to be done. One open IND has developed with hematopoietic stem cells and lentiviral therapy in humans. It involves expressing the factor VIII gene specifically in platelets. So going into stem cells with a lenti factor VIII, transducing those, allowing them to then differentiate into a variety of cell types, but putting a promoter on the factor VIII gene that allows production of factor VIII only within the mature platelets. This approach has been shown in a number of animal models and currently has an open IND. Unfortunately, with this kind of an approach, room needs to be made in the bone marrow for the transduced gene-edited cells to go back in and establish a niche. And that means that a cytotoxic agent, such as busulfan, needs to be used to wipe out part of the bone marrow. There are concerns with that in terms of a risk-benefit ratio, and so this needs to be proposed and planned very cautiously and slowly to be sure that harm is not induced in the patient from ablating part of their bone marrow. If we come back to AAV and look at some of its shortcomings, I've described the need for vector improvements, needs that need to address largely the efficiency, variability, durability of AAV therapy. I've talked about alternative delivery of genes, non-AAV delivery, such as lipid nanoparticles and lentiviruses that are emerging, and at least for lentiviruses, shortly should be in clinical trials. And then there is cell therapy combined with gene therapy, combined with gene editing, that also looks as if it can be very effective in providing for new factories, new cellular factories to produce factor VIII or factor IX, but has not yet made a transition from the preclinical models into clinical concepts. Thank you for your attention.